Okay, let's wait for another 2 minutes, then we will start. Vidasri, what specifically are you asking in lesson 8? Yashaswini and Siddhesh, your attendance is being recorded automatically, okay? There's no need to type. Vidasri, can you please specify what are you asking? Good evening, Gargi. Vidasri, I'm waiting for your text. Vidasri, are you there? Good evening, Sahan. Vidasri, which command? You have to specify what do you want to ask. I'm not getting you. Which command? Vidasri, which command are you talking about? Yes, Vidasri. So you have to specify to me what exactly are you asking, which command are you asking about? Okay, so I'm sharing my screen with Asri. You can text me later on WhatsApp, okay? What is your doubt? Okay, with Asri, you can text me later on WhatsApp regarding your doubt. Vidasri, what you have typed here, protein sequence is equal to sequence record and then the protein sequence. Vidasri, as you know, protein sequence is the variable, and here you are using sequence record, and inside the sequence record you save some attributes like ID, description, and the sequence. Right, so here inside the sequence record you are just typing the sequence. You have to save it inside those attributes. Yes, Zilam, we will think about it. Vedasri, are you understanding what I am saying? Okay, Vidasri, kindly see that lecture again. We talked about attributes, how you will store some attributes inside sequence record. Okay? Saw that lecture again. Okay, Vidasri, then I will take your doubt at the end of the class. Okay? So till now, we have worked mainly with DNA sequences. We learned how to read FASTA files. We learned how to read GenBank files. But DNA is not the final product. DNA ultimately makes proteins. So today we will learn how bioinformatics handle protein data using a database called UniProt. So what is UniProt? UniProt is a database. Just like we worked with NCBI and now we know that NCBI is a database. In the same way, UniProt is a database that stores protein sequences along with detailed information about them. So it does not just store amino acids, it stores knowledge about proteins. So UniProt is a database which specifically stores protein knowledge. So UniProt is one of the most important protein database in bioinformatics. And UniProt stores annotated proteins. Now, what do you mean by annotated proteins? So here are two terms. First term is raw protein and the second term is annotated proteins. So as you can see in the example here, this is a protein sequence. As we have a DNA sequence and in that DNA sequence, we call them nucleotides, we call them bases. Here, in the same way, these are called amino acids. So these amino acids which make protein sequence. Now, by just looking at this protein sequence, this sequence does not tell us much about its information. That is why it is called raw protein sequence. Now, its opposite, a protein sequence which includes its information like protein name, its function, organism, sequence length, its biological role. So, that protein is called annotated proteins. So, UniProt stores annotated proteins, not just raw protein sequences. Because scientists do not work with unknown sequences, they work with annotated proteins to understand biology. For example, disease related proteins, for example, enzymes, drug targets. So, protein annotations help researchers know what a protein does. Okay, are we clear about these two terms, annotated proteins and raw protein sequence? Okay, very good. So now let's open, okay, now let's open our Google. Here we will search UniProt. Here, this is the official website of UniProt, uniprot.org. And here we will click on here, UniProt. Now this is your UniProt database. So this database is specifically a protein database. As we know, in NCBI, you have nucleotide database, you have protein database, you have many other databases inside NCBI. But UniProt is specifically a protein database. Now, if I want to search for a protein, here I will simply type, for example, I want to search the proteins related insulin. Now I will click on the search button. Now here we are seeing UniProt has given me all the results related to the insulin protein. Here we are seeing insulin. Here it is written on insulin. So all the results are regarding insulin. And here we can see some are humans. Here we have dog. Here we have pig. Here, so all the results are related to the insulin protein. So let's go on the first search option. Here I will click on it. Now here at the top, in this section, we can see the basic identity of the protein. It is written P01308. So this is the UniProt ID of insulin. As we have learned about the sequence ID in FASTA file, we saw that there is a sequence ID. So here this is called the UniProt ID. And after that it is written INS human. So this means that this insulin is from humans. Okay, and then we have the protein name, that is insulin. We have the gene INS, same INS, so that means the protein is produced by INS gene. So we have gene here. Then we have the status. Inside the status it is written that it is reviewed, and in the brackets we can see Swiss prot. So it is reviewed by Swiss prot. So this means that this protein record, all of this protein record P01308, this protein record has been manually checked by experts. As we are beginners, so we will always trust the reviewed entries. If you are working with unreviewed entries, that means that most of the unreviewed entries are mostly computer generated. Okay, so we will always work with the reviewed entries. Then we have organism here, that is Homo sapiens. Here it is written amino acids, that is 110. So this means the protein length is 110 amino acids. And then we have the annotation score. This annotation score, it tells us how complete and how reliable the information is. So here we have a score of 5 out of 5, which means the information is highly reliable. Now, let's move on to the function section. So here, this section explains what the protein actually does. So let's read the first line. Here it is written, insulin decreases blood glucose concentration. So this means that the insulin helps control the blood sugar level, and this is why insulin is so important in diabetes, and this information here, it is called protein annotation. So all the data that you are seeing here, it is called protein record. All of this data. Here's name and taxonomy. You will scroll down, you will find out a lot of data. So all this data is called protein record. And it all belongs to the single protein, and that protein is insulin that we searched for. Here we can see its structure as well. Let's scroll down a bit more. Here you can see its structure as well. Let's see, here it is. So here is the structure of this insulin protein. You can search for it, you can explore the structure, you can explore the other data. Now what are we going to do today? We are going to download this protein. So to download it, what we will do, here we will select it. You have the option here. We will select it. We will click on the download option. Then we want to download the FASTA file. We already know what a FASTA file is. So we will download the FASTA file. Here you can see you have many other formats to download this file. So here we are selecting the FASTA canonical. So here canonical means the main protein sequence. Some proteins have multiple forms. As you can see, there is isoform written here. So as some proteins have multiple forms, so here canonical means the standard and the most common format of the protein. Okay, so we are going to download the FASTA format, and we want to download only this one protein file, and we don't want the compressed format. So here I will select no, and then we will click on the download option. Okay, now as we can see that this file has been downloaded. Now we will go to the file explorer section. Here we will go to the downloads. Now I will cut this file from here and I will paste it in my folder. Okay, this is my folder in the VS Code, so I will paste it here. Okay, let's open our VS Code and let's see if this file is showing there. Let's open VS Code. So here we can see our file that we downloaded from the UniProt is available here because I pasted in my folder. Okay, now I will rename it to a much shorter name. So let's rename it to insulin protein dot FASTA. Okay, so I renamed it to insulin protein dot FASTA. And as we can see that the FASTA file always starts from this symbol, this greater than symbol, and then we have the ID, and then we have the description. So this is called the header line, and this is our protein sequence. So the FASTA file is the same, but this time in our FASTA file, we have a protein sequence. Now we have already learned how to read a FASTA file, whether it contains a DNA sequence or whether it contains a protein sequence, it does not matter. We have already learned how to read a FASTA file using Biopython sequence IO module. So now what are we going to do? We will import the sequence IO in our file. So I'm writing from bio, that means from the Biopython library, I want to import sequence IO. So here sequence IO is the module and I stands for input, O stands for output. Now in the same way that we have doing to read a biological file, we will repeat that process here. Here let's make a variable. So I'm making a variable record. Then we use our sequence IO module. Then we will add dot. Here dot is called dot operator, which means I am going to call something. So whenever we want to call a function, we add a dot. So now I want to call the function read, and then we add parenthesis. So here read is a function, that's why we have added parenthesis, because inside this parenthesis we are going to give our input because we want to read this file. So we are going to give the input of this file name. Okay, so to call a function we always add dot. So dot means inside this. So inside the sequence IO module I have called the read function. Now inside this I will give my input of my file name. So here the name of my file is insulin protein.fasta, and we write our file format. So the file format is FASTA. Here we can see our file format is FASTA. Now we can print all the attributes. We can print ID. So to print I will write record.id. Now I can print description. For that I will write record dot description. Now let's also print the length of the sequence. So for this I have called the length function. Now I will give my input here. So here I am writing my variable name record and dot sequence. And we can also print the sequence as well. So let's print sequence record dot sequence. Now let's run our program. So here in the output, we are seeing that first we printed the ID. Here, this is the ID. Then we printed the description. We learned that everything after the symbol is called description. So that's why ID is also printed in this section here, along with the description. And then there is the sequence. And this is the length of the sequence, that it is 110 amino acids, the same that we just saw in the UniProt database. So are we clear how to download a FASTA file from the UniProt? Okay, so now I'm going to give you a name, a protein name, and I want you to download that file, the FASTA file format from the UniProt. Can you do that? Okay, so Sarthak, if you will download the compressed structure, then you have to uncompress it. Okay, let's download the compressed structure as well. Here, let's select, it's already selected. Let's select it again. Click on the download button. Here, it's already selected FASTA canonical, one file and the compressed format. Yes, let's download it. Now see, it is zipped. Okay, you can see in the file. This was the file earlier that we downloaded. It was uncompressed. Now this one is compressed. So to use this file, you have to uncompress it. Okay, so let's go to our downloads. Here you can see this is the compressed file. To uncompress it, you have to uncompress it. Here is the option. So you have to uncompress. You can't use a zipped file like this. Okay, that's why I downloaded it in the uncompressed way. Okay, yes. Okay, so I'm writing the protein name here that I want you to download. So I want you to download the keratin protein from the UniProt database, and in the same way that we learned how to read a FASTA file, I want you to read that FASTA file. We have already done this, I think 2-3 times, how to read a FASTA file. So just download this file in the FASTA format from the UniProt database. Okay, Lakshya, are you done with reading the file as well? After downloading the file, you have to read the file as well. Also, kindly mention your name in your program. Yes, so we have already learned how to read a FASTA file, and we have also performed this in the worksheet, in the assignment, and in the test as well. So now I am going to give you 3-4 protein names. Let me write it. First one is keratin, the same. Second is albumin, and third one is myosin, and the last is actin. Okay, wait a minute. Okay, now what I want you to do, I want you to download all these protein files in the FASTA format from the UniProt, and I want you to make a file, a FASTA file here. Make a FASTA file here and paste all these sequences in that file and then read that file. Are you getting what I just said? You have to, first of all, you have to download these proteins in the FASTA format. I have shared 4 protein names. You have to download all these proteins from the UniProt, and then after downloading all the files, you will have different FASTA files. One will be of keratin, one will be of albumin, one will be of myosin, and one will be of actin. Then what you have to do? You have to create a FASTA file. Just like we create a Python file, for example, I write it lesson 11. So I'm writing here lesson 11, the name of my file. To make it a Python file, I will add the extension .py and then I will press enter. So this is how I make a Python file. To make a FASTA file, I will write .fasta. Okay, then it will be a FASTA file. So you have to create a new FASTA file. Now in this FASTA file, you will save all those 4 sequences and then you will read that file. We already learned how to read a FASTA file with multiple sequences. In that case, we do not call the read function. In that case, we call the parse function and we use a loop. I have pinned the message here, Zililam. This is keratin. Okay, I have written the wrong spelling here in keratin. It is Keratin. There is no O. Okay, the keratin, albumin, myosin, and actin. These are the 4 proteins. Okay, let me write it again. Wait a minute. Someone was, yes. David, you were asking about how to download from UniProt. Here, look here, David. You have to open your Google. Then you have to type UniProt here. And then you will see the official website of the UniProt, uniprot.org. You have to click here. So this is the UniProt database. Here you will type your protein name, for example, actin. And then click on the search button. Here you will see all the results relating to this protein. And then you have to download one protein in the FASTA format. Okay, to download it, for example, this one, here you will click on this, you will select it, then click on the download option, then you will click on no. You are not going to download the compressed format, you are going to download uncompressed format. Okay, so that's why here select no. So this is the FASTA canonical. As I said, the canonical means the common FASTA, the common format of the protein. Okay, and then you will click on the download option, your file will get downloaded. David, is it clear? Okay, so the message is pinned here. Okay, let me unpin it. Here the message is pinned here. So Deshna, you already know what loop I am talking about. You have performed the question many times, Sudeshna. Yes. So you remember, right? We learned 2 ways how to read a file. One, if our FASTA file only contains one sequence. So in that case, we call the read function. If our FASTA file contains multiple sequences, for example, if I copy it and paste it here, so now our FASTA file contains multiple sequences. Now in this case, you are going to call the function parse here and you are going to use the loop. Okay, we have already done this earlier. Yes, so very good. Download all the proteins. Here I have pinned the message and just like I said, make a FASTA file in your VS Code, paste all those sequences in your FASTA file and then read that FASTA file. Choose the other one. You have many options, choose the other one. Okay, Aditya? David, David, see here. David, see, if I go into this file, here I am seeing the download option, right? If we are going into the file, suppose this is the file, here, this is the download option. Now, if I click here on the download and I select the FASTA canonical, this is the entry, and I click on download. So this is how it is showing. This is your screenshot. Okay, David, this is your screenshot. So that's not how I told you to download. You need to go back where you selected this file to go inside this file. Okay, so you need to go back, select here. You have the option to select this. You have all the options to select how many files you want to download. So select this file and then here click on the download. Okay, and then click here no, and then the FASTA canonical, and then click on download. Yes, Gargi. Sakshi. Okay, David, are we clear? Sakshi, this is not how I told you to read the file. Very good, Siddesh. Sakshi, look at the Siddesh code. The last screenshot in the group. Sakshi, I want you to make a FASTA file. Click here on the new file option. Write your file name, for example, actin dot fasta. Okay, if you want to make a FASTA file, you have to add dot fasta at the end, just like we are seeing here, dot fasta. So this will make a FASTA file. Then after creating this FASTA file, paste all the sequences in this FASTA file and then read this file, actin dot fasta. Okay, you can give your file name anything you want. Sakshi, are we clear? Okay.